I did my research on the difference between Clorox and method products. I chose this experiment because my stepmom, who's here, likes to use method products, which is an all-natural cleaner, and it's better for people with asthma and allergies. It's a little bit less strenuous on the breathing. Clorox I chose because my mom uses that, and it's an all-chemical cleaner, and this is man-made. And to see the differences between which one will kill the bacteria more efficiently. I chose door handles because a lot of us, we go through our daily routine, going in and out of classrooms, grabbing the door handle, and then touching our phones, our face, mouths, whatever. And I thought it was interesting to see the bacteria load that is found on these door handles. So like I said before, Clorox is an all-chemical cleaner. So what this means is they are man-made and they are manufactured in a laboratory. So the bacteria this claims to kill is Staphylococcus aureus, the Escherichia coli 0157H7. This is a virulent type of the E. coli bacteria. This is what you hear on the news when there's a food outbreak and can make people very ill. We also have Salmonella entericia, Streptococcus pyogenes, and Klebsiella pneumoniae. These range from normal flora, which means they are normal bacteria that are found on your skin and in your intestines, to something that is very pathogenic and that can cause a lot of health concerns. So we also have the method products. These are the all-natural antibacterial cleaners. And what that means is that it is all-natural in the sense that it uses citric acid, which is found in like lemons and other fruits and vegetables. The bacteria that this claims to kill is Staphylococcus aureus and Salmonella entericia. So these are a little bit less, it kills a little bit less bacteria than what Clorox claims to kill, but also they both are able to kill Staphylococcus aureus. So here are the three types of bacteria I was testing for. And just as common knowledge that I feel like everyone should know is that S. aureus and S. epidermis, which is the normal flora on your skin, can last 2 days to 12 months on hard surfaces, and I found this at my resource by what we know. The three bacteria types are Staphylococcus aureus, Staphylococcus epidermis, and E. coli. The Staphylococcus bacteria are the gram-positive, and the reason for that, you see here, they are colored purple, and the reason is because they have a thick peptidoglycan layer, which picks up the stain, the dye, when you're standing on a slide. The gram-negative, however, the E. coli is going to be pink, and that's why it's gram-negative, because the peptidoglycan layer is thin, and it's covered by an outer membrane that does not allow for penetration of the dye. So I wanted to look at this because I think it's important that everyone knows that there's a lot of bacteria found on many different hard surfaces. Luckily for us, I was dealing with non-pathogenic bacteria that was found. This can only cause problems if ingested, or if you have a cut or abrasion on your skin and the bacteria from your normal flora gets into that abrasion and can cause problems. For example, if somebody has a serious surgery and the bacteria somehow gets in, it can cause a bone infection, which then would lead to hospitalization. There are pathogenic or virulent bacteria that can already cause this. So you have MRSA, VRSA, and the E. coli 0157H7, which I talked about earlier. MRSA and VRSA are not able to be killed as easily by antibiotic treatments. And so MRSA is methicillin resistant, and VRSA is vancomycin resistant, which are very strong antibiotics used in the medical field. And luckily none of that was tested, so that's good for us. So my hypothesis was that the all-chemical cleaner, which is Clorox, would kill more bacteria than the all-natural cleaner, which is method products. So my procedure, I decided to swab the door handles of the chemistry lab and the microbiology lab. The chemistry lab, I used method products, and the microbiology lab, I used Clorox products. And you see here that on the left-hand side, I swabbed the front of the door handle and behind it, right where somebody's hand would rest. And then I would plate it onto TSA plates, which is tryptic soy agar. So Clorox on the microbiology door handle, I would clean it and I would leave it sit for 10 minutes. And then method products I would leave on the chemistry lab door handle for 4 minutes. The reason why is because these are the directions that the manufacturer suggests to disinfect a hard surface. From here I used TSB, which is tryptic soy broth, and from each of those broths that I made 9 new plates of each of the following. So TSA plates, MSA, and EMB. I also tested on Mueller-Hinton plates, and each set of plates and broth I incubated for 48 hours before I went and looked at them and then conducted more studies. So here are my main results. As you see here, it's a very small bacteria load, but what I worked with, and I'm going to use the microbiology before that was using Clorox as my example, with the 4 colonies and the 3 different colors. So here you see I have the plate, which is the big white, and then I have 4 different colonies. There's 3 different colors. From each color I took and made a broth, and from each broth I made 3 new plates. I used TSA as my control. This allowed me to make more bacteria growth if need be, so I could test it further. The MSA tested for gram-positive bacteria, and this was your Staphylococcus aureus and epidermis, and the EMB was testing for gram-negative, which is your E. coli. So this is my overall data. So on the TSA and MSA, everything grew. Nothing grew on the EMB, so I did not find any E. coli at that given time when I swabbed the door handle. I did, however, find both Staph epidermis and Staph aureus bacteria. And this is also my overall results. So you see here the tan is the TSA, which is your normal media that you use just to grow. And then these 2 select for, this is the MSA, and that selects for the positive, and this selects for the negative kind of bacteria. So overall, I had 2 Staph aureus bacteria that grew on the MSA, and then I had 7 S. epidermis bacteria that grew on the MSA. These are the normal flora I was telling you about that are on your skin and just on surfaces, and usually don't cause a problem unless it gets into a cut or something of that nature. Like I said before, there was no growth on the EMB, so there was no gram-positive bacteria. And then I tested for Mueller-Hinton to further test for any antibiotic resistance. And I'll get into that in a minute. This is my overall. I just used the same chart of my before and after, my before, because the afters, nothing grew after I cleaned it and re-swabbed the door handle. So you see here that from week 1, I only grew Staphylococcus epidermis, and week 2, I grew Staph epidermis and Staph aureus, and chemistry lab before was the only one that grew Staph epidermis, Staph aureus. My results here with the Mueller-Hinton plates, this was further tested because of talking to a professor and him suggesting that I test for the antibiotic resistance. So I used triple sulfa, ampicillin, and nafcillin. We usually would use vancomycin, but at the time I did not have it. And we just used this to see. And again, test for antibiotic resistance, and luckily no growth that I saw at the current given time. Which, that doesn't mean that it can't grow anywhere else or any other time. So in conclusion, Clorox and method products killed the same bacteria, and that's what I was mainly looking at, was the bacterial load and if it was killed off, and it was. And my hypothesis that Clorox would kill more was incorrect. And I would like to thank Dr. Hilwig and Dr. Stein and Cheryl Maurer, the professors that helped me along with this process. Also, Corinne Igley, who is the student assistant that would help these professors, and Marissa Loretto for taking these awesome photos of me. Any questions? Yes.
What does the university clean with?
I'm not too sure, actually. That's the only thing that I was not able to find out, I know the cleaning ladies come in once a week. They come in on Fridays, but I don't know what type of product they use. I would assume some type of cleaner that would kill the bacteria, but I know probably not Clorox or anything like that. Just knowing from my mom working at a school, they use, it's like a heavy duty, but I don't know exactly the brand.
All right, thank you, Briana.